Hello everyone, my name is Kevin Kemp and my presentation is on systemic lupus erythematosus and the human body. Systemic lupus erythematosus, known as SLE, is an autoimmune disease that affects multiple systems throughout the body. This disease is difficult to diagnose because there is no clear cause. There are many risk factors involved with the onset of SLE, both environmental and genetic, such as mutations to the genes, or factors such as UV sunlight exposure, cigarette smoking, alcohol consumption, and even contraction of the Epstein-Barr virus, known as infectious mononucleosis or the kissing disease. In SLE, the body's immune system targets its own cells and tissues as if they were pathogenic. So the body is reacting negatively to its own tissues, causing inflammation in a lot of areas, which is one of the main problems in patients with SLE. Women are 9 times more likely than men to develop SLE, and their onset is usually around the ages of 15 to 45 during their childbearing years. During those times, and usually around the onset of SLE, there's a lot of hormonal changes going along with the individual, and due to the close linking between the immune system and the endocrine system, this could be a potential reason and area for research. African American women are affected at 3 times the rate of Caucasian women, and they're also more likely to experience more severe symptoms of the condition. So it was important for researchers to understand just how these risk factors play a role in disease development and disease expression in the form of those severe symptoms. After an individual is diagnosed with SLE, they experience a drastically shortened life expectancy. And unfortunately, SLE cannot be cured. Treatments work to manage symptoms and prolong the lifespan. Many treatments available are more antiviral medications or some even anti-malarial medications. One notable one being hydroxychloroquine, used to treat symptoms in patients with SLE. There are many symptoms and criteria of SLE, and this heterogeneous presentation is one of the leading causes and difficulties of diagnosing an individual with SLE and coming up with a clear criteria of development of the condition. Some of the major symptoms of lupus include a sensitivity to the sun and ultraviolet lighting, joint pain, any problems with the kidney function, constant fatigue or tiredness, having sudden weight loss or unexpected and drastic weight loss, rashes, especially seeing this type of malar rash pictured here, where it's a butterfly rash that goes under each cheek and around the bridge of the nose. The pain, the SLE individuals will constantly kind of have a low-grade pain around them. Some of the criteria for lupus is the malar rash, a discoid rash, which is like circular rash, photosensitivity to UV rays, oral or nasal ulcers, arthritis, serositis, so infection of the serous membranes, renal disorders, neurological disorders. Some patients with SLE can even have such severe symptoms as experiencing seizures from the condition. A hematological disorder, so the blood not functioning properly or having proper levels of each chemical, and blood particle formed elements, presence of like anti-DNA and anti-cellular antibodies within the individual, and also having those ANA, which are those anti-nuclear antibodies, which target the individual's own DNA cells. Now, this individual must have 4 of the 11 criteria, so any 4 of these conditions or criteria can lead to a diagnosis of lupus. So with all of these varying conditions, it makes it very difficult to have a clear criteria as to what causes this to develop, if the presence of any 4 of those can be determined as an SLE diagnosis. So in the immune response, cells are constantly undergoing apoptosis or necrosis and releasing nuclear particles, which are exposed to the immune system for prolonged periods of time in an individual with SLE. As you can see over here on this diagram, where apoptosis occurs, and then a phagocyte will come and eat these apoptotic bodies, so they are not in the extracellular environment. However, in SLE affected individuals, these phagocytes do not fully uptake all of the particles, allowing some of these nuclear and intracellular particles to be exposed to antigen-making cells in the immune system. Now, some of these phagocytic cells are neutrophils, and when neutrophils are released, they have NETs, NETs, which are neutrophil extracellular traps made out of their own DNA and histones, which end up exposing more intracellular autoantigens into the environment. And neutrophils also trigger other immune responses, such as more B cells to come out, which leads to higher inflammation levels that can damage organs. T cells are activated, which activate B cells to target the body's own intracellular components for destruction. So again, more activation of these B cells, which are targeting the body's own DNA, which usually respond to foreign pathogens only. B cells develop antibody receptors that are specialized to attach to antigens, which are usually found on pathogens. However, an individual with SLE has production of antibody receptors that are designed to target their own intracellular particles. And these B cells are targeting the fragments left by cell death and apoptosis, which we visited earlier. And because those phagocytes are not uptaking all of the intracellular fragments like they should be, that prolonged exposure in the extracellular environment affords the opportunity for these B cells to attack and create an immune response on the own DNA of the SLE infected individual. And when this happens, it creates immune complexes that promote inflammation and attract other immune cells to fight off what is the believed infection. Except when these immune complexes are produced in large numbers, they cannot be phagocytized properly, and these prolonged periods of inflammation occurs, and especially in an SLE affected individual where they already have an issue with phagocytizing cells properly. This can be a problem. This diagram is a quick recap of everything we've discussed, how these initial risk factors such as mutations, UV light exposure, or Epstein-Barr virus can lead to increased rates of cell death, which lead to other immune responses such as phagocytes, T cell activation, which all relate to B cell activation, which form immune complexes, which then lead to inflammation in specific tissues without the body. And these are some specific areas where you can get it, such as the dermatitis of lupus, which would be where the rashes, and glomerulonephritis in the kidney. And these are the references for my report, and thank you for your time.